Good evening, everybody. So my feeling in the last few days is that there is a lot of information coming from the media, and it's sometimes a bit difficult to evaluate the data to see what is accurate, what is not, and what is important and what is not. So today I think I try to summarize relevant facts about the new coronavirus and give you my opinion, at least, for how I see possible therapy or other means to stop this virus will develop in the near future, hopefully. So the overview, the outline of my talk today would be to discuss a little bit about coronaviruses in general, talk about the history of coronaviruses, which is quite long. We'll review the current epidemiology of the disease. I will touch briefly on the virology aspects of this virus. And then I will devote some time to talk about the putative drug targets that we can use in order to stop this virus and other therapeutic approaches. And as Zohar mentioned, we'll have some time for questions and hopefully also answers at my lecture.
Join the meeting.
Coronaviruses, actually we're talking about a large family of viruses. Here in this region you can see what is called a phylogenetic tree, showing only a handful of viruses from this large family. This is the new virus that emerged and we're talking about today. This is the initial name that he got before the current nomenclature. I will briefly mention other viruses from this family, but you can already see that all of those viruses belong to what is known as the beta group of the coronaviruses. There are other groups, there are alpha and also gamma that are not shown here in this tree. Now, if you can see here in this micrograph, this is a negative stain image of coronaviruses. And from looking at this micrograph, you can actually understand the origin of the name, because here you can see a haze of a white density. This white density belongs to the spike protein of the virus, which is very important protein that is used, and it's very important drug target, and I will discuss about this spike protein. But you can see that it surrounds the viruses and gives them an appearance of a corona. This is the source of the name. Also, it's important to mention that we're talking about envelope viruses, meaning that they have a membrane coat around them. They use RNA as a genome, which is also important. And we're talking about a single piece of a genome in a length of about 30 kilobases. So those are the coronaviruses. Now, a little bit about the history. So the first coronavirus was actually identified in the 60s. And this coronavirus was detected as a virus that infects chickens. And the name of this virus is avian infectious bronchitis virus, or IBV. And it's still circulating globally. It is considered as an agriculture problem. And you may have heard in the news, there is a research institute here in Israel who developed a vaccine against this particular virus. The institute is called Migal, and now they're trying to convert their vaccination approach to combat the human virus. Also, for quite some time, we know about other coronaviruses that infect humans. Here you can see an example of two of them. There are additional ones, and they typically cause common cold symptoms. Sometimes pneumonia, but no more than that, so they're not lethal, and are also globally distributed. Now, in 2002, a new coronavirus emerged that was in China, and the name of this virus was severe acute respiratory syndrome virus, or SARS. It emerged in November, but it lasted only until July, the middle of the summer, and this will become important. I will mention that toward the end of my talk. So this was quite a lethal virus. There were about 8,000 cases, almost 800 death-related cases, which gives a mortality rate of almost 10%. And the economic burden was quite significant. And you can see it here, it was estimated to be around $50 billion. Of course, we estimate the current situation to be much more costly. So that was the first emergence of a really deadly coronavirus. And then there was another one in 2012 in Saudi Arabia. This virus is known as the Middle East Respiratory Syndrome virus, or MERS. So far, it's caused 2,500 cases, and the lethality rate is actually quite high. We're talking about 34%. And this virus is still circulating. So you can see this graph, it's still circulating, still infecting people up until now, but the spread of this virus is very slow, and I will also touch about that in a few slides. And a more recent history, in December 2019, in the Hubei province in China, a new coronavirus emerged, and it got different names. At first, it was known as the 2019 new coronavirus. Now we call it the SARS coronavirus 2, and we call it like that because it's really similar to the previous SARS coronavirus that emerged in the early 2000s. And this is probably the way we picture this global pandemic, many people wearing masks and fearing the virus and trying to not get infected. Now, a little bit about the epidemiology of the virus. Here you can see data that I pulled from, it was true for this morning, but unfortunately, it's changing very rapidly, so by the hour. And here you can see the number of new cases reported by the days. And in blue, you can see the reports in China at first, and then in the rest of the world. And you can see that in China, the situation is mostly resolved. They have it under control. There are no more new cases almost. But you can see the huge spike in new cases in other places in the world. Now, a little bit about the mortality rate that was debatable, at least for this virus. If we look at the number of cases, so I think we already passed the 200,000 cases as of now, but in the morning it was still 194,000 cases. And look at the number of the death cases, then we're talking about a mortality rate or fatality rate of about 4%, which is quite high. But this is an average fatality rate, and we see huge differences if we look at the different countries. So unfortunately, in Italy, we know that the situation is very severe, and the fatality rate in Italy today is almost 8%. Unlike Italy, in other cases like South Korea, they managed to somehow suppress the mortality, and you can see that they managed to suppress it down to 1%, which is significantly different from this 8%. And mostly it's due to the ability of the healthcare system to admit new patients and to give them intensive care for a long time. We know that there is a shortage in Italy in intensive care units, and this is probably why we see such high mortality rates. But if patients are able to get proper care, then the mortality rate is significantly lower. Also, it is quite clear now that there is a very strong age relationship between the mortality rate of this virus. And you can see that it's much more severe for older people. And when we talk about young adults or kids, we see very low mortality rate. This is very untypical. It's not something that we used to. So if you compare that to flu, for example, in flu, we also see higher mortality rates for the younger group of people, for young kids. We don't see that with this new virus. And I think this is one of the more encouraging aspects of this pandemic, because as a society, we always care about the kids. And I think from these data, it seems like the kids are fairly safe with this new pandemic. But of course, on the other side, we have the older people that are in severe risk from dying from this virus. Now, additional epidemiological data. The onset of symptoms for half or 50% of the cases are at 5 days. But up until 14 days, we see this spread. I don't know if you can see that in this graph. And from that, all the governments are basically deciding on their policies. At least here in Israel, I'm not sure about other places. We have 14 days of quarantine for people that were exposed to the virus. And this was made under the assumption that for people that didn't develop any symptoms up until 14 days, they are probably not infected. Now, how people know that they are actually infected, we can see the symptoms that this virus is causing. So the highest percentage is shown here. So most of the people develop fever, and they also develop a dry cough. And those are the most prevalent symptoms. There are other symptoms that are more common for other upper respiratory tract viruses like nasal congestion and other things, but you can see that there are lower rates of such instances. And currently, people that have fever and they cough, it actually might be this particular virus. Now, a very important aspect of viruses is how they actually spread in the population. And there is a number that is derived from computational models that are fitted to the data of the patients, trying to predict basically from each patient how many other people might get infected. So this is the reproduction number that is shown here, or R0. And you can see that different viruses have different numbers. So one would be a linear growth. Everything below 1 will tell us that the virus is decaying. Everything above 1 will tell us that the virus is spreading and expanding, and the rate will depend on this number. So for Ebola, for example, we're talking about numbers at this range, about 2. Influenza that people are familiar with, we're talking about roughly 1.5, sometimes 2. Depends which influenza virus we're talking about. Measles is the most infectious virus that we know about, have very, very high numbers. And if we look at the estimated numbers for this virus, we see that it's anywhere between 1.5 to 4, meaning that it's more infectious than a typical seasonal flu. And this is something that is important to consider when we talk about this virus. Okay, let's switch to talk a little bit about the virology of the virological aspects. So here you can see a schematic diagram of the genome of the virus. So we're talking about an RNA virus, meaning that it's a single-stranded RNA virus. It's a positive sense, meaning that the virus, the genome of the virus, can be translated as soon as it enters the cells, and it starts to produce the proteins. Now, there are several proteins here that I would like to highlight because those are drug targets. So there are 2 specific proteases. Proteases are proteins that know how to cut other proteins. So there are 2 proteins here, the PL-PRO and 3CL. Those are proteases, very unique proteases that the virus is bringing with him. And their function is to cut this very long polyprotein that the virus is making. So a polyprotein is one very long polypeptide chain that is need to be cleaved in those highlighted regions in order to produce the functional subunits. Now, if we will inhibit those proteins, of course, the cleavage will not occur and the virus will not be able to propagate. So those are very important drug targets. Also, there is the RNA-dependent RNA polymerase. All RNA viruses need to bring some kind of a polymerase that knows how to read RNA and replicate it. Otherwise, they won't be able to replicate using the cell machinery because normally we don't have such polymerases in our cells. So this is another very, very important drug target. And the last one that I want to mention is this S protein. This is the spike. This is the glycoprotein that appears on the cell surface, on the surface of the viruses. And this is, of course, very, very important target for vaccine design and antibodies, and we'll review that in a second. Okay, how can we stop this particular virus? So we can think about different things. First of all, we can think about treating the virus itself, treating infected individuals, of course. And from that, we can either use off-the-shelf drugs that might be available, and we'll see which ones might actually do that. Or, of course, some specific drugs that will be developed against this particular virus. Now, if we talk about large-scale prevention, then the only real method of doing that is vaccination. So we'll talk a little bit about vaccines. And then there is another intermediate usage of different drugs, and this is for prophylactic treatment. Now, prophylactic treatment is given before people get infected in order to prevent infection. The thing is that it could practically be done for a small group of people that are at high risk, like medical care providers, for example. So we'll talk about that also. So let's start with off-the-shelf drugs. As I told you, the RNA-dependent RNA polymerase is a very important protein that the virus brings with him in order to replicate its own genome. Here you can see the structure of this protein from the previous SARS, the one that infected in the early 2000s. And you can see that structurally, it's very similar to other RNA polymerases coming from very, very different viruses. And this is not a coincidence. This is indicating to us that the function of this protein dictates a very conserved appearance, a very conserved structure, and that gives us an opportunity to target those proteins. And many people have tried to develop inhibitors, specific inhibitors that will inhibit those proteins. And I will mention a few of them. So I want to mention ribavirin. This is probably the most used drug against any viral infection that no other treatment option exists. So for many years it's been used. It's not highly effective, but it has some benefit. And of course, it was also used in this pandemic. I would also like to mention this molecule. It's called remdesivir, and it was developed by Gilead Sciences, initially to target Ebola. And also we'll see the effect of this molecule on this virus. And I just want to mention that there are other drugs. Some of them are shown here, and there are many more that people have developed over the years. Now, a very encouraging paper was published 2 or 3 weeks ago, actually in the middle of February. And this is a paper that is summarizing a long research that was done in non-human primates, in which this particular drug, the remdesivir, was tested against MERS. MERS is the Middle East Respiratory Syndrome virus, very similar to the virus that we are now facing. And here you can see that there is actually a benefit of using this particular drug, both for treating monkeys that were infected with MERS, and also to prevent infection when this drug was given in a prophylactic manner, meaning before the actual infection. So that gives us a lot of hope that this drug will also be useful for treating the current virus. Now you can see here a testing of a few compounds against the virus, against the new SARS coronavirus already. You can see ribavirin, which is not very effective, that is shown here. And here you can see remdesivir. The numbers that we typically look at are those numbers. It's EC50 telling us what is the concentration of the drug that we need in order to inhibit 50% of the virus. And you can see that it has a very low number, meaning that the drug is actually effective against this virus. I also want to mention here another drug, chloroquine. This is not an inhibitor of the RNA-dependent RNA polymerase like all of the other drugs here, and it functions in a completely different mechanism, but that I won't describe now. But you can also see that it's quite effective. Now, chloroquine is a known malaria drug. And the good thing about this particular drug is that it's widely available. So in every pharmacy that you will enter is likely to have chloroquine available to treat malaria. Unlike remdesivir, that is experimental drug, and it's still not available in the market. The pros of using those kind of drugs is that those are immediately available. Some of them have shown low efficacy in the past. It could be better in this case, but we still don't have data for that. I just want to mention that there are several clinical trials that already started with remdesivir. We're still waiting for results, and until then, we won't know for sure if it's really effective against the virus in treating patients. Some of them have high toxicity, some of the compounds that I'm showing here. As I mentioned, there are two proteases that the virus is using, and are absolutely necessary in order for the virus to replicate in cells. And those are excellent drug targets. And people have already developed multiple inhibitors against those proteins, and then developed them against the proteins that were actually derived from the first SARS coronavirus, the one that emerged in the early 2000s. Now, we know that the proteins of the current virus and the proteins of the previous SARS are very, very, very similar. In fact, in the active site, they are identical. So all of those compounds that were previously developed against the old virus will be effective against the new virus. The problem with those compounds is that they never reached any clinical testing. So they were developed, and that's it. So those would be a very good starting point to further develop better inhibitors. We can improve them, but they will need to be evaluated in clinical settings, something that was not done so far. And of course, that requires a lot of time. So the last thing that I want to mention in respect to off-the-shelf drugs are antibodies. In recent years, we have more and more positive experience in treating various pathological conditions with antibodies, starting from cancer and ending in infectious diseases. The target of such antibodies would be the spike protein. And here you can already see the structure of the spike protein of the new SARS coronavirus. So this is the target of those antibodies. Now, the biggest question would be whether or not we already have antibodies that will be able to target this spike. Now, in order to enter cells, the viruses need to bind something that is known as the cell entry receptor. And they do that by using this particular domain that is shown here, and the cellular receptor of SARS coronavirus, both the previous one and the current one, is a protein that is called ACE2. It has a very important role in regulating blood pressure. Hence, there is a relationship also with the higher risk of people that have a high blood pressure. And this is an excellent target for antibodies that will be able to bind and neutralize the virus because they will prevent it from binding to its own receptor. Specific antibodies that were isolated against the previous SARS are not very effective. You can see here in those straight lines, basically they don't bind the new SARS protein. Nevertheless, we have anecdotal indications now that some antibodies might actually be effective. So this is an example for one of them that was now very, very recently identified. So it was originally isolated against the previous cells. It binds it very well, and it retains some binding to the new cells. So it's a very good starting point, maybe, to develop this antibody further. And also animals that were vaccinated with the previous cells develop antibodies that are also effective against, or to some degree effective, to the new cells. So it gives us hope that we might actually find antibodies from these pool of antibodies that were previously developed against the old cells. Also, I want to mention Takeda. It's a Japanese company that have a technology to isolate the antibody fraction from plasma of people. So they're taking plasma samples from people that recovered from the current cells, and they're starting to produce that, and that would be another potential drug that will be available soon. So basically, that would be a concentrated antibody fraction from plasma of people that recovered. And it's supposed to be effective against the virus, of course. So we talked about off-the-shelf drugs. Now let's switch to vaccines. What can we do about the vaccine design? So, of course, vaccine design is a very, very active research field. And here in this table, I'm showing you, it's a table that is taken from a review that was published a few years ago about efforts of people that try to design vaccines against the old SARS, the previous SARS, that made use of the spike protein. So all of those vaccination approaches made use of the same protein, but you can see that we have very, very different approaches, like taking the full-length protein and injecting that, taking a DNA that will encode this protein. And in this respect, I can mention the company from the US that announced that they have an mRNA technology. So basically, they have mRNA that encodes this particular protein, and many other technologies that people have used. I don't know why I have a drawing on my board. Okay, so be it. Anyway, one of the platforms that I personally think would be very useful in fighting the disease is shown here. Basically, it's a class of platforms that make use of viruses that are harmful to humans, they're harmless to humans, and we can engineer those viruses. So for instance, this is an example for a vaccine that was developed against Ebola virus. So the VSV is one virus that we can engineer. We can take the spike protein of the Ebola virus, we can introduce it into the VSV virus and create this chimeric virus. So that was done. It was actually done by a company called Merck, and it's now known as Irvebo. It's a vaccine that was licensed very, very recently. That was 2 months ago only. And you can imagine that we can take the spike protein of the current SARS virus to put that on either VSV or other viral platforms that were developed and produce vaccines that will look like that. The very good advantage of these vaccination approaches are that those viruses are replication capable, meaning that you can inject them to people and they will replicate. They will not cause a disease, but they will replicate. And by their replication, they will enhance the immune response. And in this regard, we actually analyzed the immune response of people that got this particular virus that was developed against Ebola virus. And we isolated, in collaboration with a group in Germany, we isolated antibodies against the spike protein of Ebola virus, and we characterized the way they bind, the way the immune system see the virus or the vaccine. And the conclusion is that this particular approach really presents the viral components in a very ideal way that is suited to develop a very good immune response. So this is one reason that I think that this approach would be one of the more promising approaches to fight the coronavirus. So many groups and pharmaceutical companies already started to produce such vaccines. And the question is, what would be the roadmap for development? So as I told you, the manufacturing already started, but the first obstacle would be to show that those vaccines are actually advantageous, and that could be shown in some kind of an animal model. There are different models that people already started to develop. It could be as simple as using mice. It could be more complicated as using non-human primates. But it might actually be more challenging. We still don't know that. Once this would be shown in animal models, then we could approach or proceed to clinical trials, but that will take time. So building or creating vaccines, that would be the best method to achieve control, but that will definitely take time. I don't know exactly how much, but it will take time. I can assure you that there are very, very strong efforts now to proceed as fast as possible. So we talked about off-the-shelf drugs and vaccines. Now I want to refer to specific drugs against this particular virus. One of the approaches that seems to be very promising in recent years in fighting various different viruses is to use monoclonal antibodies. And I also think that once we'll have good monoclonal antibodies against this SARS coronavirus, that will also be a good treatment option for people. Now, the roadmap for developing or for isolating such antibodies is well known. It's a bit complex. I'm not going to go through all the different steps here. But this is something that was developed in recent years. There are different groups and different pharma companies that know how to do that. It involves some characterization that takes time. This is mostly where we do some, we contribute to this effort in the characterization step. And once we'll have antibodies, then the roadmap for using them is looking like that. So we'll need to isolate them, we'll need to characterize them, and then we'll need, like vaccines, to test them in some kind of an animal model and then proceed to a clinical trial. However, antibodies are known to have a very good safety profile, unlike experimental drugs. And potentially we can actually get approval to use them for compassionate care protocol, and this is basically a very fast protocol to allow the usage in cases where there aren't other means to treat people. And this is the protocol that is now being used in order to provide possible therapy with remdesivir under the compassionate care protocol. So once it's known that it's not toxic in humans, you can actually approve the use of such experimental drugs to treat patients that don't have other good options. So the benefits of antibodies are that they are very safe, they're very effective, and we can actually do that quite fast. I think that would be much faster than developing additional small molecule drugs like the inhibitors of the proteases, as I previously mentioned. However, using antibodies would be far more expensive. So producing antibodies is expensive, and if we think about mass usage of such antibodies, that actually might be a problem. Before I end, I just want to mention that there are other approaches, and I want to mention our own research in this respect. So potentially we can also produce something that is called an immunoadhesion. Immunoadhesion is a molecule that on one hand has some domain that will allow it to bind the target. In our case, we're talking about the cellular receptor of the virus. This is the ACE2 protein. So potentially we can take this ACE protein and fuse it to another portion of an antibody to create a chimeric moiety that will be able to bind the virus on one hand and recruit the immune system on the other hand. So this approach is something that we are currently pursuing in my own lab, and we hope that we'll be able to come up with something promising in the near future. So with that, I would like to end with this more optimistic slide. So in general, it's good to relax. And I selected this particular slide because it shows you summer. And we know that upper respiratory tract viruses, or all the other respiratory tract viruses that we know about, are attenuated during summertime. So the infection rate of those viruses reduces. There are less instances, and there are great hopes that this would also be the case for this particular coronavirus. And if so, at least at summer, we'll have some alleviation in the severity of this pandemic in the northern hemisphere, at least. And it will give us more time to prepare toward the next winter that will eventually arrive. And with that, I will open the stage for questions and answers. And so thank you very much.
Thank you so much, Ron. Everybody is on mute, but I can say on behalf of everybody that we really appreciate and we really thank you for the information. During the discussion, there were some questions coming. Some of the questions are repeating themselves. So I will try to bring them all together into a few categories of the same question, sort of the same question. One of them is, can you have any comment on the results obtained in Italy with a use of tocilizumab, a monoclonal antibody for rheumatoid arthritis, which was tested in Italy according to the question that came from Italy, by the way, from the northern part of Italy. And the question is if you have anything about this specific thing. And another question that came regarding Italy is the virus in Italy is basically already a mutation of the corona.
Okay, so it would be easier to answer the second question. So as far as we know, the answer is no. We don't see, or at least we don't have any data that supports the existence of quasi-species that have different infectivity of the coronavirus. Of course, those RNA viruses accumulate mutations. And we see that, we know that there are mutations, but we don't have so far any evidence that there is a selection for a particular mutant that could basically indicate that it has any advantage or that it's causing more severe spread of the virus or anything like that. So I think the answer currently is no. As for the first question, I'm not aware of those trials. I just want to mention that the amount of data that is piled is enormous. There are literally hundreds and hundreds of different research papers that appear now. It's actually quite difficult to keep track of all the things that are going on. And also to really look at the things that make important contributions on the background of all this general noise in the field. So I did not hear about this particular monoclonal antibody. I can only guess that it's something that was designed to block the ACE receptor. And if this is indeed the case, I don't know what is the outcome, but in general, it's an approach that might work. We'll need to see data.
Thank you. A few questions regarding the vaccine are related to the timetable. of development of it. And basically the question that is how long will it take until the vaccine will be really effective for humans? We hear a lot about a year and a half from now. Is it that so long till we will be able to really use an effective vaccine against this specific virus?
Okay, so first of all, we don't know exactly how long it will take. It might actually take a year or more.
Ron, if you can take the sharing of your screen off, it will be, I think, easier for everybody.
Let's see if it works. Okay.
Super, thank you. Sorry,
Okay.
again, sorry for disturbing you. Regarding the vaccine time frame.
Okay, so unfortunately, yes, it will take a long time to get a vaccine. How long exactly, we don't know. The bottlenecks are testing the vaccine. It's not the development. The development was done already. There are many vaccine candidates that already went into production by various pharmaceutical companies. Now they might start to test for toxicity in parallel of testing their efficacy in animals, but or they might actually try to gauge their effectiveness in human subjects that will take part in the phase 1 clinical trials. Basically, they can try to take blood samples and see if they develop antibodies that are capable of neutralizing the virus. But that will not provide information for the ability of the vaccines to really prevent infection, and only clinical trials will be able to tell us. And unfortunately, those trials are typically very hard to conduct. There are not the typical clinical trials like treating people. So treating is very easy because you can enroll people that already have a condition that you want to treat. The vaccination is designed for people that are healthy and have certain probability to be exposed to the virus. So typically we're talking about clinical trials that are much more complex, much more involved, and much more expensive and large than any other clinical trials. But I don't know, there might be exceptions, there might be some regulatory exceptions in in the face of this pandemic. So if those will take effect, we might have vaccines a bit similar. But I want to stress that also rushing forward is not always a good idea.